Hi class, and welcome to your next video on cells. So we've talked a lot about macromolecules, but now we're gonna move into the cells and what those macromolecules might do for the cell. So let's first introduce exactly how did we find cells. Now in 1665, Robert Hooke first observed cork from plants under a microscope and coined the name cell. He actually looked at it and he thought it looked like a monk's cell and a monastery, and that's where we got the term cell. At the same time, Antoine van Leeuwenhoek observed pond water under a microscope and found all kinds of living things, and he actually called them the term animalcules. And they were actually little microorganisms, and a lot of them, what we know as bacteria, is what he was looking at. So this is the cork under a microscope, and as you can see, it's very, very rigid actually, and it has a very good structure. And if you can think back on what Robert Hooke might have thought, you can kind of see how he might have thought it looked like those monk cells in a monastery, and this is how we probably got that term of cell as we know it today. So Schleiden and Schwann were two very important men as well. They came up with what we know as cell theory. Cell theory states that all living things are composed of cells. Cells are the basic unit of life, and all cells come from pre-existing cells. Now, if we think back to our characteristics of life, this makes sense because we said that cells were the first unit of life in the hierarchy of life. And so all living things have to be composed of cells. And that is our basic unit of life. And now we know that all cells must come from pre-existing cells. So we have two main types of cells, and we'll get further into these types in a minute. But the first type is a prokaryotic cell. It's small and simple, and it has no nucleus, but we call that a nucleoid region instead. And that's where it has a circular DNA chromosome, so it does have DNA. It has no membrane-bound organelles, and what that means is it doesn't have a membrane around its organelles. It does have organelles, but it does not have a membrane around it. This is all bacteria. It has ribosomes, which means it's going to be able to produce proteins, and that's going to be very important for expressing its genetics. It has a capsule around it, and it does have a cell wall as well. Now, eukaryotic are a lot larger and a lot more complex, and the nucleus, it has a nucleus, which is where we store the DNA, and it has membrane-bound organelles. Like I just said, that just means we have some membrane around our organelles that are going to do some kind of job or purpose within the cell. Additionally, they can be unicellular organisms or multicellular organisms. Over in the prokaryotic, we only find them as unicellular organisms. Now this here is a pretty good resource for us to look at how big or small a cell is. So if we look at this here, we see a coffee bean, a grain of rice, times regular 12-point font, and we can see that we're at this is 1 millimeter. Now, if we go in a little bit, we see that sesame seed, and it's 3 by 2 millimeters, and we can see a grain of salt is 0.5 millimeter. But then we go in even further, and an amoeba proteus is a eukaryote, and it is 500 micrometers. That's our first prokaryote that we're going to see, or eukaryote that we're going to see. And then we go in a little further and we see a paramecium, and that's another eukaryote. And then we see a human egg, which is another eukaryote. And we start seeing how big and small these different actual cells are and the general size of these cells. And if we go in even further, we see the skin cell. Your skin cells are only 30 micrometers. Then we can see a red blood cell, which is even smaller, at 8 micrometers. And then we get in here and we see what is the E. coli bacteria. The E. coli bacteria is 3 times 10 to the 0.6 micrometers. It's a lot smaller than that egg cell or even the skin cell. So we're actually seeing the different sizes in the prokaryote versus the eukaryote. So the prokaryotes are a lot smaller than the eukaryotes. And this is actually the mitochondria that we're going to talk about in a minute that is within your eukaryotic cells. Okay, even within eukaryotic cells, we have plants and animal cells that are 2 different types of cells. And there are big differences between them. In the plant cell, they're both eukaryotic, but the plant cell contains a large central vacuole, which is going to contain a lot of fluid, and it is also going to contain macromolecules for storage. It's also going to contain a cell wall to help provide support around the cell. It's going to be a very rigid cell. And the plant cell is also going to have chloroplasts to help create sugars. The animal cell contains small vacuoles, but not a very large one like the plant. But it contains no cell wall or no chloroplasts. Now the cell parts. Cell parts are called organelles, like little organs, and they all have functions. So let's go through them. The first one is the nucleus. The nucleus has several parts. The most important role of the nucleus is to house the DNA. It's like the instruction book for the school, or for the school, or more importantly, the cell. The nucleolus, with the darker part in the nucleus, is the ribosome factory, and the nuclear membrane is the outer covering of the nucleus. The chromosomes are tightly wound DNA found in the nucleus, but we also have unwound DNA, and that's called chromatin. We have cytoplasm, and that's what all of our organelles are going to sit in. It's similar in consistency to Jell-O. It's a very watery type consistency, and it just kind of holds everything in place there. You'll also hear it referred to as the cytosol. So it's just that matrix that everything is going to sit in. Okay, we have protein-making parts. Proteins are really important because this is how we express our genes and we express our DNA. So the first one is the ribosome, which is the site of protein synthesis. What that means is we're making proteins at the ribosome. These can be free-floating in the cytoplasm, or they can be attached to the rough endoplasmic reticulum. The rough endoplasmic reticulum, the one that has the ribosomes attached, makes proteins destined for secretion. It is studded with the ribosomes, making it rough. The smooth endoplasmic reticulum, the one without the ribosomes, is in charge of lipid synthesis, metabolism of carbohydrates, and detoxification of any toxins or poisons. Modification and transport of those proteins. So we're going to make all the proteins at the ribosome and at the rough endoplasmic reticulum, but then we need to modify them. We need to proofread them. We need to make sure they are perfect before we use them. So we're going to receive proteins from the endoplasmic reticulum in a transport vesicle, and proteins are modified as they move through the Golgi apparatus. So we're going to kind of just look at them, and we're going to make sure they're usable and there's no big mistake that's been made. And then the Golgi is going to ship them off to the final destination, another destination somewhere in that same cell, or it could be another cell completely. It receives it, it modifies it, and packages it, much like the post office. And then the transport vesicles are like the mail truck that delivers the protein somewhere. So we're going to be delivering it, we're going to modify it, we're going to make sure it's ready for use. So every now and then something bad happens and things break down and the cell is not going to be up to par. So we need to recycle things within the cell. We need to recycle these organelles. So that's where the lysosomes come into play. Lysosomes are full of digestive enzymes which break down food and old worn-out organelles of the cell. And we're going to help recycle old parts as well. So it's going to digest these old organelles and we're going to recycle them. And we're going to reuse the parts that we can reuse. So lysosomes serve as the cell's digestive system. Lysosomes contain enzymes that break down individual macromolecules or even whole cells. So we just digested that food with the lysosome, so what are we going to use it for? Well, cells need energy, and we're not going to actually get energy from the sugar or from the fats or from any of our macromolecules. We're actually going to need to get it through ATP. So we have to convert it. And the way we convert it is we send those digested materials to the mitochondria, which is the powerhouse of the cell, basically. And the mitochondria take sugar and turn them into ATP. This is a useful form of energy, and it's also called adenine triphosphate. And once we take and we transfer that into ATP, we're then going to be able to use that energy. So you might be thinking, okay, animal cells have mitochondria, but don't plant cells make their own food? Yes, they do. But they don't make ATP. They make sugars through chloroplasts, and they go through the process of photosynthesis. So chloroplasts in the plant cells convert sunlight into sugars. Sugars are only used for structure and storage without breaking down. The cell cannot use it for energy. So the cell must use the mitochondria to convert the energy to ATP. So all cells that are eukaryotic cells must have a mitochondria. All plant cells must have a mitochondria. Lastly, cells have to move. So cilia sweep back and forth like a broom, and there's going to be a lot of them. And flagella, you might have one or two, are going to be using in a whip-like motion. Okay, make sure you've got great notes, great pictures, and we're going to talk about this in class.